So there is evidence of vaccines, cancer vaccines, that function as primary preventers of those diseases.
Elias, thank you for coming. You are a PhD and professor of neurosurgery and pediatrics, as well as principal investigator of the RNA engineering laboratory. You are the co-leader of the UFHCC Cancer Therapeutics and Host Response. So you are, in my mind, a cancer researcher and a doctor at the same time.
Correct.
Right? So you treat patients, and not only you treat patients with available wealth of knowledge that the medicine community possess, but you're also trying to expand that wealth of knowledge, right? You're trying to find new methods, new avenue, like new ways of curing people. Yeah. So, Elias, maybe we can start with cancer itself. What is cancer, right? How is it different to normal cells in the body, and how does it relate to other types of cancer? Because as you mentioned, you are a brain cancer doctor and researcher.
Absolutely. So cancer is very different from normal cells. Often people describe cancer as normal cells that have changed or a disease of self. And I've really struggled with those definitions because cancer is not like normal cells at all. Cancer is also not just abnormal growth. If you just had abnormal growth, that could be what we might refer to as a benign tumor, or a tumor that's growing very discreetly and can be just cut out. One of the hallmarks of cancer, though, is it actively infiltrates and damages the local architecture of tissue, wherever that cancer is. So as an example, in the brain, one of the most common brain tumors, malignant brain tumors, is something by the name of glioblastoma. Glioblastoma grows like this, tentacles that are infiltrating the brain. And as that tumor tentacles its way through the brain, it is damaging the normal architecture of that brain. And that's very difficult to treat because you can't just cut it out, because these are cells that are infiltrating in all of these directions.
Is it tentacle-like because it's constrained to grow in that way, or just because glioblastoma is different from other cancers that do grow like that?
It's complex.
Okay.
Cancer is literally creating an environment, an ecosystem that is really most suited for its survival. So when these cells first start growing, they're actually dying because the environment is so challenging for even cancer cells to survive in.
Is it the immune system already attacking it, or just the environment is hostile to its existence, something like that?
The immune response is involved.
Right.
In many cancers, early on. In the context of glioblastoma, again, the most common malignant brain cancer, the immune system starts to see the cancer not as a foe that it has to fight, not as an enemy, but rather as a friend or a wound it has to heal. So that is the big challenge with the immune system. The immune system is not, most people think of it as just a killer force. And it is, but the immune system has another role. And the role of the immune system is also in the context of healing. You cut yourself, you injure yourself. The immune system is involved in healing injury. And so in the context of glioblastoma, a lot of brain cancers, the immune system is really just trying to heal this very challenging damage response that is taking place. And it actually becomes part of the problem because as it tries to heal the damage, it allows the cancer to invade and destroy. And so it becomes a vicious cycle. The cancer is invading and destroying. The immune system is trying to protect it, heal it, and that allows the cancer to invade and destroy all the more. And this it's just a vicious cycle upon itself.
Is it trying to, how does it help? For example, is it trying to heal the surrounding cells around the cancer, or in a way it's trying to help, as you said, like it's a friend, of the cancer cells themselves?
It does both, to be honest. It can actually directly form a shield around the tumor, preventing good cells or good immune cells from fighting and killing the cancer. It can also directly provide crosstalk to tumor cells that enables their proliferation, their survival. Much of this is still trying to be understood in terms of the connectivity between these immune cells and these tumor cells, and the crosstalk between not just the immune cells and the cancer cells, but the immune cells and other immune cells and other cells in that environment. But to make a long story short, these are complex environments. And it's not just one cell turning into 2, 4, 8, 16, 32, 64, et cetera, et cetera. We're talking about fundamentally an ecosystem that the cancer is most suited to survive in. And in this ecosystem, this doesn't look at all like the normal environment. Immune cells enter this environment and they're shocked. They literally, and many of these cells actually go into shock, the good immune cells. The words we use is they become anergic. Anergic means no energy. And so this is an environment where the cancer dominates. It's an acidic environment. It has low oxygen. It's often quite acidic. It's very difficult for normal cells to live in that environment. And even the cancer cells, many of them don't survive in this environment. So if you look at glioblastoma, the center of the tumor is what we call necrotic or dead. And so the cells that are the most evolved, the cells that are most responsible for the local regional destruction, they're the ones not just surviving, but migrating away from this death, invading the tumor, invading the normal brain.
Is this ecosystem something that is different from other types of cancer? So like, does this, because I think it's called the tumor microenvironment. Am I correct? Okay, so is this ecosystem
very good?
I try to do my homework.
You did.
So is this different? Does that exist in other types of cancer?
Absolutely. Okay. Absolutely.
Maybe what ways is brain cancer different from, well, you've already said, but like, is it fundamentally this? It's fundamentally the same thing, right? Like, that's kind of what I'm trying to answer. Like, I'm trying to question is the process that's happening in brain cancer different from other types of cancer?
I would take it a step even further. Mm-hmm. I believe the environment of every cancer is unique to the individual.
Right. Okay. So,
and we can start to say maybe brain tumors are generally categorized by this environment, and bone tumors are categorized by this environment, and lung tumors, et cetera, et cetera. But generally speaking, if we're talking about cancer, it's an ecosystem that is uniquely personalized, an environment that is uniquely personalized. And in this environment, you have unique metabolism. Mm-hmm. You have...
And personalized, I'm sorry, you mean to a specific person, right?
Correct.
So like different people, even though they have the same exact type of cancer, will have different environments, right? Something
exactly.
Like that.
Exactly. Yeah, that's exactly right. I mean, this is... In my experience, every one of these is truly its own unique entity. Okay. And that's become, at least in my opinion, one of the grand challenges of how do you create a therapy that serves an entire population when even within the same disease, you're talking about a disease that literally is an N of 1, meaning it is the person who has that's the only one of its kind.
It's like patient serotype, almost analogous, something like that.
Yeah, so it becomes very hard to say, oh, okay, this is what we learned in this one person's cancer. We're just going to apply it now to everyone else who has the same cancer. I think you can do that to a certain extent, so long as it's appreciated that these really are very different across individuals. And I'm not even talking about the genetics of the tumor, which are different from patient to patient to patient. Mm-hmm. I'm talking about even the environment of that tumor.
How it connects to the other cells, how it interacts.
Exactly.
How the immune system interacts and all that kind of stuff.
Exactly.
Okay. So maybe it goes to the question that I asked you before. could you, for example, as a brain cancer doctor, treat or like have a good amount of idea of what to do with a patient that has other types of cancer, something like that?
I think it helps to be a pediatric oncologist. So I'm a pediatric oncologist. I take care of children, adolescents, young adults. The work we've pioneered in the lab, though, has been used in adults, and hoping to start treating children very soon with this approach, children with brain cancer. What I will say, though, is as a pediatric oncologist, we're taking care of patients with what we refer to as orphan disease. And by orphan disease, I mean it doesn't affect enough people that you would say it's common. And so it's often referred to as an orphan disease. This is a little different from very, very common cancers in this country: lung cancer, prostate cancer, breast cancer, etc. However, going back to our understanding of cancer now, which is it's an entirely unique and individualized disease, an N of one, so to speak. With that definition in mind, every cancer is an orphan disease. Every cancer is unique to an individual. Right. And so to answer your follow-up question, what we have learned in taking care of patients with orphan disease, I do think can provide a blueprint for helping us understand cancer more writ large, and providing a template for how we treat patients, which I think has to be more personalized.
Right. And I'm definitely going to ask you about that when we get to the vaccines, because I've had this idea that vaccines can be like personal or universal.
Exactly.
And we'll talk about that. But for example, for brain cancer specifically, you mentioned prostate cancer, lung cancer. For example, it is, I think, common knowledge that, like, for example, smoking correlates or causes tumors in the lungs, right? How does the brain tumor start? Is there like something that we can say, oh, maybe this has caused, or like it's very correlated to brain cancer, or is it just something that happens?
Yeah, it's a great question. I will say this question is probably the question I get asked most frequently by patients, care providers. Right. And it's the question that I must answer most poorly.
Okay.
And the true answer is we don't know. Right. And that's the real answer. I follow that up often by saying it is likely a complex process that we don't fully understand of an individual's genetics, how those genes interact with the environment, the environmental triggers that, again, may be unique to that individual based on their own personalized genetics, and how that leads to the development of cancer is likely very much a crosstalk that we don't understand, but involves these things, genetics, environment. And it's more than just genetics, it's epigenetics, meaning how genes are turned on, how genes are turned off, which is especially true in pediatrics, because pediatric cancer is not really a change in the genes, which happens in adults if there's a mutation, from smoking, et cetera. Mm-hmm. It's usually what we refer to as epigenetic, or genes that are supposed to be off that turn back on, like developmental genes that are important for rapid growth during when a baby is developing in utero. Those genes have to be turned off, they get turned back on, and some of these pediatric cancers can develop.
Is this a thing that's more common with children, or not necessarily?
No, cancer is certainly more common in older patients. Okay. It's certainly, cancer in general is more common in older patients.
Body starts to break down more, and that breaking down leads to mutations. Maybe.
Exactly. As cells age, they're more prone to errors, especially if there are certain environmental triggers. Some that we know very well: people who sit in the sun and don't put sunscreen on, UV radiation that can lead to skin cancer. Certainly, alcohol, smoking, a lot of lifestyle acquired changes, viruses, for example, can also lead to these types of things, cancer. So, but in children, it is uncommon. But when it happens, it's often thought of more as an epigenetic or genes that are there that are supposed to be off that turn back on again. Whereas adult cancer tends to actually be a change in the gene.
What are the we'll start slowly going into how do we treat this. So, but for example, for these kinds of people, what are the types of diets that they have to eat? Is there something specific that they have to eat when they're dealing with glioblastoma, for example?
It's a great question. So diet is something that has been studied extensively in the context of cancer, and we still don't have great recommendations. I can tell you what we know generally. What we know generally is cancers often use sugars as their source of primary fuel. So people have looked into starvation of the tumor by cutting out sugars almost entirely. While that can help, cancer is nothing if it's not evolution. It is constantly evolving. Meaning you introduce a therapy and it's trying to evolve, find a way to resist it. And so
I guess with any cell, right? But just in this case, we're trying to get rid of this specific type of cell. So it's kind of like I'm under more pressure to figure out ways of surviving.
Exactly.
Something like that.
Exactly. And so starving it of sugar often puts pressure on it to find another means of using energy, using fuel, using food. And so really the treatments that work, and I think you're familiar with them, are these very crude approaches. Poisons like chemotherapy, radiation, burning it, cutting it out completely. That's why they work, because it's such a complex process that you almost have to hit it with these very, very significant therapies to really get all the cells all at once, very quickly. Yeah. Because if you give it a chance to evolve, if you give it a chance to escape, it can be very, very difficult to eradicate. And so while these treatments, in terms of diet, can have merit in terms of improving outcomes, they're very challenging to lead to a significant effect on their own.
Right, you have to bring a holistic network of solutions, right?
Correct.
And one of the solutions, for example, we can start talking about them that you mentioned is chemotherapy. You mentioned the word toxic, which in some ways is kind of surprising because it's the common way of treating cancer, and calling it toxic is kind of whoa, right? But at the same time, I think I know what you mean, right? Maybe could you explain what toxic is and why do you specifically mention that word, right?
Well, when we think of the birth of chemotherapy, much of these agents were warfare agents once upon a time, or developed from the knowledge of developing kind of poisons, toxins in the context of warfare. And yes, of course, it's evolved into a treatment, but the idea is these are poisons. When nurses are handling these, they're handling them with appropriate gloves and inhalation standards for many of these agents, and they were injecting them intravenously into children. So these, you'll make no mistake, these are poisons. These are toxins that are designed to destroy cells that are dividing quickly.
Could you go over a little bit more specifics? What is chemotherapy? I imagine there's many types of chemotherapy, but maybe you could talk specifically for brain cancer at the very least.
Yeah. Generally speaking, these drugs that we refer to as chemotherapy are in a class of agents, class of drugs that disrupt the cell's ability to clone itself. So all of our cells need to divide. They have to divide to maintain themselves. And some cells grow a lot faster than others. As an example, hair grows faster than some of the other normal cells in our body.
Other hair in the head, hair in the feet, or something like that.
So hair on the head tends to grow pretty quickly, and that's one of the reasons why these quote-unquote poisons can disproportionately affect the hair follicle, meaning the hair falls out very quickly when patients are started on these chemotherapeutic agents designed to disrupt the cell from dividing. even faster than the hair follicles division is cancer cells. They are dividing very, very, very quickly. And so they are very sensitive to chemotherapy and can die very quickly in the setting of these drugs. But these are not targeted to the cancer. Right. These chemo drugs, at least the most common ones we use across cancer, flood the human body and affect all the cells. Right. Just the ones that divide very quickly tend to be more disproportionately affected. And the cancer cells are the ones who are the most affected if they're dividing very, very quickly. Now, for some cancers that are more slow growing, that's harder. It's harder to get the same effect. Mm-hmm. Or they have subclones that are what we refer to as senescent or stem-like or don't grow and don't populate, meaning you get rid of all the rapidly dividing cells. And this is why a lot of patients recur. They have a population of cancer cells that just sit there and don't change.
Mm. And they're still
and then they
damaging.
Grow out.
Right, like
they are these quote-unquote stem cells just kind of sit there often. Okay, so and sometimes they can renew themselves, but many of these cells are kind of, senescent or indolent or just kind of there, but not doing a whole lot.
Right, so and not then, necessarily dangerous.
Dangerous in the sense that they can wake up, and when they wake up, they can populate the entire tumor again.
There's like a possibility of it'll come back and it'll be damaging later.
Absolutely. And so how do you get rid of those cells? That's very challenging with chemotherapy if they're just kind of not being driven into a rapidly dividing phenotype. So standard of care for cancer really involves what I would refer to as a multimodal, multi-pronged approach. Surgery, chemotherapy, radiation, really hitting the cancer with everything that we have to prevent that one cell from surviving this and repopulating again.
Maybe you can go a little bit to the surgery extraction, so like the cutting, basically. Like it literally means open the brain, go find the cancer and just scoop it. Yeah. Something like that.
Basically.
Right. So, and what are the possible negative effects of that? Like, why would you not want to do that necessarily?
So it's a standard of care, and there are risks to surgery, of course, especially surgery in the brain. But I have to say, for cancer, it is one of the most important treatment and curative options. It really is. However, when you go into the brain and you try to remove these tumors, there are some tumors that are not even amenable to surgical resection, depending on the location. So if a child has a tumor in the brainstem, you can't take out the brainstem because the breathing is controlled by the brainstem.
So by taking the cells, you're likely also taking the other important vital cells?
Exactly. So the location of some tumors makes complete resection impossible. But there are other areas of the brain where you can completely remove the cancer. The problem is, if it's a benign tumor, you can cure it with just surgery. But if it's not a benign tumor, if it's a cancer that's infiltrating, as I described, removing it removes what you can see, but it doesn't remove what you cannot see. Right. And if you don't remove exactly, right. And those are there, and those have to be treated with these other modalities like radiation, chemotherapy. And in the old days, people literally would take out did horrible things like take out the entire side of the brain to remove what they thought was glioblastoma. And it would grow back on the other side, which is really proof that this is the microscopic disease that is infiltrating the entire brain. And so in this context, this very aggressive cancer, like many aggressive cancers, need what we refer to as a systemic treatment that goes everywhere and can eliminate these cells.
What are other alternatives? For example, T-cell therapy a completely alternate, different way of treating as the technology that you're developing?
It's similar. So immunotherapy it's a form of immunotherapy. Right. It's a form of immunotherapy. And immunotherapy has gained a lot of traction over the course of the last 15 years. I will say immunotherapy has a long history, 150-year plus or so history, going back to the late 1800s of people trying to use the immune system to fight cancer. But really, I would say only in the last 15 years have we really proven in randomized phase 3 clinical trials, really proven that it works. And so there are many different forms of immunotherapy. T cells are one of the most promising approaches. The problem is, it's complex therapy. Getting someone's cells out of their body, engineering them to fight their cancer, reintroducing them, it's complex.
Right. And we're also already getting into a little bit of the details. So maybe how does immunotherapy work in general? And why would you want to use immunotherapy? Because, for example, maybe I read this somewhere, one of the reasons that cancer is so devastating to the body is because it weakens the immune system, correct?
It does, yeah.
So then that's, for example, when we hear it's like, oh, my grandmother had cancer and she didn't die of the cancer necessarily, but like a flu that she caught while having cancer, something like that. So how does that work?
Yeah, it's a great question. So you're absolutely right. Oftentimes patients with cancer have a very much weakened immune system. One of the major reasons why they have a weakened immune system actually is the treatment, chemotherapy, for example.
Okay. Right.
Chemotherapy, because it can destroy cells that divide rapidly, some of the cells that divide the fastest are immune cells. And when immune cells are dying in the face of chemotherapy, then a virus, a bacterial infection, a fungal infection, which otherwise would never cause an issue, can be fatal in a patient who undergoing active treatment with cancer. But it's more complex than that. The cancer itself can actually affect how the immune system works. This is certainly true for glioblast, going back to our example of cancer.
Like the tumor microenvironment, right?
The tumor microenvironment, the way it reprograms the immune system, subverts the immune system. This is not just in the tumor. This has effects on the whole body. And so cancer is not it's not just a local disease. Okay, again, a benign tumor, local disease. Right. But if you're talking about cancer, this is a body-wide disease.
Right, that happens to be in like a specific location, but affects the whole body.
Absolutely. And the immune response, certainly in the context of glioblastoma, is very much affected. These cells have a very difficult time in doing the normal things they can do. Things like even trafficking, their ability to move from where the factories where they come from to the site of infection or the site of the tumor, that becomes very much compromised in the context of a tumor which may be located in the brain. So, this is very complex biology. Cancer, it's biology that is complex in the tumor with its own ecosystem and its own environment. It's also complex biology in terms of the systemic effects, the effects this has on the entire body. And they are profound. And this involves many organ systems. One of them is the immune response. And the immune response is very much adversely affected in the face of cancer.
The immune response, when cancer starts to grow, the immune response in a way knows that this is something that it's not supposed to be there.
Correct.
So
Yes.
It starts attacking it already, right?
Often, yes.
Okay. So
not always, but often, yes.
Depends on the type of cancer.
Perhaps. I think it's more individualized, but yes, generally speaking, correct.
Okay. So immunotherapy. One of them is T cell therapy. One of them is, would you characterize it like vaccines, mRNA or not? If there's the other way is that if that's being treated for brain cancer, are those two different categories?
I consider all of these in the class of immunotherapy.
Okay. And how does? Yeah, maybe we'll go into details, right? Like what does that mean, right? Like it means that you somehow helps tell the immune system to attack the cancer maybe more aggressively than what it would normally do, correct?
The whole idea of the immune response, the immune system, is the immune system should know friend versus foe. Mm-hmm. So when a baby is developing, any immune cell that recognizes oneself is eliminated. And everything that's left over is literally the incredible versatility of the immune response to respond to anything foreign. It's truly incredible. I mean, human biology is absolutely incredible. So fundamentally, every patient's immune response should be able to fight their cancer with that premise in mind.
Right.
Which is why I often struggle with this idea that cancer is a self-disease. It is not. I understand why people say that, but cancer is not normal. And the immune response absolutely should have the versatility, the biology, to kill every cancer. And I would argue that it probably does, and we're just not aware of it.
Okay.
We're aware of its failures. But the idea that it can be resurrected in the fight against cancer is true, because now we have agents that we know can resurrect, can give the immune response the help it needs to destroy cancer and clear it. Now, it's still early days, because immunotherapy still fails more than it works across all human cancer. But now this premise is there. It can be used. It can be resurrected. And so people often who are referring to cancer as a self-disease, I often respond with, okay, if cancer is a self-disease and the immune response can't recognize it as foreign, how does the immune response recognize our normal cells infected with virus? Because the immune response is killing normal cells infected with virus. That is proof that the immune system has this incredible versatility. That one of our normal cells infected with virus may look 99.9% normal, with the exception of now the virus inside of it. And yet the immune system can recognize that and destroy that. So why not cancer? And I would argue a cancer cell is far more different than a normal cell infected with virus.
Okay. So how do we tell it that it's, or you are a researcher that is studying vaccines to help or in a way make stronger the immune response, right? How does that work? And maybe we can now talk into the details specifically of your specific research, right? There's very interesting aspects of it, like, for example, that it's an mRNA vaccine. And another one, the way that you introduce it into the body, like with the lipid nanoparticles and the structure that it has and all that kind of stuff. And maybe we'll start with the mRNA. Like, why? What does that mean? Like, that it's an mRNA vaccine, and what exactly it's doing to your body that then affects the cancer?
Yeah, so there are many different types of cancer immunotherapies, all designed to resurrect the immune system in this fight. You mentioned T cells. People are trying to take T cells outside the body and reprogram them against their cancer.
And T cells are a specific type of immune cell.
They are the part of the immune system that, so when I gave the example of the virus, the immune response can recognize that normal cell. It's T cells. It's T cells specifically that do that can recognize those normal cells that are even slightly different and get rid of them. So this has been known for a long time, which is why people have focused on T cells. How can we use T cells, resurrect T cells in this fight? Other approaches that have been developed are giving drugs to the human body that can take the brakes off these cells. Maybe they're there. Maybe they're activated against the cancer. We just need to give them an additional push. And so we're seeing these drugs.
Like medicine for the immune system?
Basically. There are these drugs that are commercially available now, Keytruda, Opdivo. These are drugs by the generic names are pembrolizumab, nivolumab. They basically, they're antibodies that target the brakes of these cells. And by taking the brakes off these T cells, they're much better fighters of cancer. And that seems to work very well in adult cancers like melanoma and lung cancer that have a lot of mutations. So basically resurrecting these cells in the fight. But they don't work all that well, I would say, in most cancers because a a lot of these cancers don't have a strong immune response to begin with. So giving them these drugs, you're not boosting a whole lot that's there in the first place. But it's a good place to start in the sense that we now have proof that the immune system can be manipulated in certain ways to achieve cures in patients who largely failed every other therapy before that. So that's a great starting point for us to advance new research, new technology. My own research focus, yes, has been in the mRNA space. The term vaccine or cancer vaccine is a bit of an oxymoron, as you might imagine, because when you talk about vaccine, you're talking about preventing something before it happens, and that's largely not what we're talking about. Right. We're talking about
something already exists.
Yes, exactly. And so the more appropriate term is mRNA therapy that we're trying to provide patients. I do like the term vaccine, honestly, because I believe in my heart one day we will get to what that really is.
Oh, interesting, like a preventative care for cancer.
Exactly, exactly. In my heart, I believe we can get there.
Right.
We're not there yet. Mm-hmm. I do think, though, that there can be an avenue for something referred to as secondary prevention, meaning you've gotten rid of the cancer with standard therapy. Maybe you just did surgery to remove it, but you know that a patient is high risk for recurrence. So instead of giving them quote unquote preventable chemotherapy, you maybe can give them a vaccine. That's what we refer to as secondary prevention, meaning something's happened, you've gotten rid of it, but you're worried it comes back, you give a vaccine to prevent it from coming back. And I think that is a great starting point for this idea of vaccine, but even there, it's really more of a treatment.
Gotcha.
Hopefully one day we'll get to what I hope will be primary prevention. I'm a pediatrician, so vaccines I think are one of the greatest, if not the greatest, invention in medical history. Preventing something before it happens. We still have a ways to go with cancer, but there are cancer vaccines that prevent cancer from happening. One example I can give you, or actually more than one example, there are vaccines that prevent viruses that we know cause cancer. The hepatitis B vaccine, as an example. The Gardasil, which is a vaccine against the human papillomavirus. We know these work to prevent cancer in these settings. So there is evidence of vaccines, cancer vaccines, that function as primary preventers of those diseases. But we still have a long ways to go. They're few and far between, and my honest hope is we will have many more examples of vaccines that not only work to prevent cancer from coming back, but preventing cancer from happening in the first place.
So mRNA, what does the mRNA mean in this setting, and how does? I'll let you explain. Like, what does RNA has to do with on any of this, right?
So RNA, honestly, is the information highway of the human body. I mean, it's really the best way to describe it. The human body has a blueprint for itself, and that's DNA. Most people are familiar with DNA. That is literally the blueprint that makes each of us.
Blueprint is like instructions, right? Like this, I can create my human body in a sense, right?
It's the blueprint, but that blueprint has to be read. And that blueprint has to be, when it's read, right, that information has to be conveyed. So imagine there was a blueprint to build a building. Someone has to read the blueprint. And then the person reading the blueprint then has to inform the workers, here's what we're doing. And then the building comes up. The human body is the same. It's very similar. Mm-hmm. Yes, the DNA is a blueprint. But in and of itself, that's it. It just sits there. It's a blueprint. And it really requires the information to be conveyed to manufactured protein, manufactured organs, manufactured systems that do all the amazing things the human body does. RNA is literally the messenger going from the blueprint to the building, the production.
Somewhere in the body that something needs to happen.
Exactly. So the production is protein. I mean, that's us. We're protein. Our bodies are made of protein. to make that protein into its unique shapes and sizes and colors and everything that's uniquely us, it's RNA. RNA is literally the conveyor of that information. And you can imagine if the information highway of the human body is RNA, the ability to interrupt that or modify that information or can be truly transformational in terms of not just vaccines, but therapies, gene therapies, cancer therapies, turning off the immune system if you want to turn off the immune system, turn on the immune system if you're trying to turn on the immune system. It can revolutionize healthcare as we speak. And the reason I think it's so revolutionary is not just because of what I said, but honestly because of how easy it can be to deliver personalized care in a way that's commercially amenable, in a way that you can centrally distribute. So what do I mean by that? When the SARS-CoV-2, COVID-19 pandemic came about, many people were really leery of the idea that we could develop a vaccine for coronavirus. And the reason that is it hadn't been done to date. Mm-hmm. And there's a very specific reason why it's very hard to vaccinate against coronavirus. It's changing.
Right. Like
so you go through the
variant,
right,
whatever variant. Right,
so you go through this laborious effort of making a vaccine, and it's different. Mm-hmm. So early on in the pandemic, many people in the scientific field felt like this is, maybe this is not the best use of resources. Mm-hmm. Maybe we should just be focusing on treating it and limiting the damage it does, as opposed to trying to prevent it. Now, people in the mRNA space very quickly felt that this is a potential solution because of how quickly you can change the mRNA to adapt and overcome the virus, how quickly the virus is changing.
And quickly, you mean in terms of time for whenever you inject the agent to change the responses in the body? And also, do you mean the time to create something like this, right? Like, because, for example, for the COVID vaccine, it was months, right? And it was fairly quick.
If you know a sequence, you can make a vaccine very rapidly. Now, centrally manufacturing it, distributing it, quality assuring it, releasing it,
right.
That was the year's time. I'll be honest, when the sequence of Spike was available, our lab made a Spike, SARS-CoV-2 vaccine in a couple of weeks. I mean, it was that easy for people with know-how to make a vaccine. Mm-hmm. Now, we didn't have the billions of dollars to do what I just said, and quality assure, release, cold chain. A lot of things had to be worked out, which took a lot of time and took a lot of research. But the idea that you can quickly make a vaccine when you know a sequence is there for RNA. And on a large scale, we have proven this to be true, that you can adapt these vaccines to keep up with COVID-19 evolution. Mm-hmm. And why this is important, it's important because the virus, it needs hosts. It needs to spread from host to host to host. So if you're staying ahead of its evolution by vaccinating uninfected people now with the new virus, with the new information, you're protecting those people and you're not allowing that virus to spread like wildfire anymore. So let's go back to cancer. Mm-hmm. COVID-19 on a large scale is very similar to cancer in an individual. It's trying to spread. It's trying to find an evolutionary escape mechanism. And it often does. And that's why we can't rescue a patient anymore. The technology isn't there yet, but imagine we can understand with artificial intelligence, with some of the boon in next generation sequencing and molecular phenotyping. What if we can understand the direction a cancer is going before it happens and actually intervene in that patient with a vaccine that prevents that scenario? that is an example of trying to contain this before the wildfire happens in the human body. And as technology improves, I think this will be quicker and quicker and quicker. I mean, it takes a long time to do what happened for COVID-19 on a large scale. But if we fast forward the clock 5 years from now, 10 years from now, 50 years from now, I see an era where a patient comes in and from a blood test, their cancer can be phenotyped or really understand the exact fingerprint of that individual's tumor. And a very specific vaccine that's unique to that patient could be available maybe even the same day. New technologies are coming out that can allow us to make RNA vaccines, RNA therapies much more rapidly. And I'll give you an example. If we were to go back in human history, one of the greatest inventions in human history was the discovery of the printing press. It's one of the greatest discoveries of the last 1,000 years. Gutenberg's type movable printing press. That allowed information that had to be recorded by hand. It allowed it to disseminate to the public, and I mean that created a revolution on its own. The knowledge now was able to be shared in real time. We, I believe, will generate technologies that allow us to take the human biology, the human body's information system, and be able to type it in a way that's much more rapid for the purposes of therapeutic delivery.
So how does that work? Do you need to have a sample of the tumor or like this cancer cells and kind of like extract in a way like its genetic sequence or like sequence its genetic code, and then with that create something that says immune systems attack this, something like that?
More or less, you need to understand the disease well enough to know what are the weak points to target.
Okay.
And then once you know that, you just need to make the information to tell the human body to do it. That's it. If a human body's missing a certain protein, or there's a genetic deficit, just deliver information to the body to make it. If the human body has an overactive immune response and you want to turn it off, information can be written and delivered that does just that. So you're absolutely right. You have to understand the disease, which is unique and it's personalized. And that's some of the challenge right now that we have. But who knows, with some of the new technologies in terms of prediction and understanding, and who knows, maybe machine learning, we can have a fundamental understanding of an individual's disease in a way we never had before. And once you understand that, once you understand the enemy, you can find the points of weakness.
Right. So maybe a point of interesting conversation is what I was mentioning before of the personal and universal vaccine, right? Like in a way, the vaccine for COVID, you could categorize it as universal vaccine, and this would be what you're envisioning is more personal.
It is.
And is that what you're currently researching, or like the work that you're doing, is it like that very personal approach, or like maybe like a step in the middle, right? Like something like, oh, we'll get like a universal, like a code of how this behaves sort of universally, and then apply it to these people. Or like you're talking with your specific approach specifically to like, okay, you, Mr. Elias, let's get your genetic, like your blood, for example, as you mentioned, to get the genetic sequence and do the best thing for you specifically.
I think you can do both.
Okay.
I really do. And I think both will be developed in parallel.
In parallel, okay.
So yes, you're absolutely right. What I described is a very uniquely personalized approach, and some patients may really warrant and demand that. But I think the universal approach is going to happen probably sooner rather than later, maybe even before some of the personalization comes together. And I do think you can universalize an approach that's personalized. Okay. What do I mean by that? Right. A lot of human cancers have shared targets, have shared points of weakness. If you screened all human cancer, why couldn't you make 100, 1,000, maybe 10,000 mRNAs that encode for shared points of weakness across human cancer. Now, the challenge is one person may have shared points of weakness 1, 8, 5, 12, 15, and another patient may have 3, 6, 7, 25, 34. But if you've made them all ahead of time, you literally pick up off the shelf the mRNAs that are unique to that individual's cancer's points of weakness. So you have it produced ahead of time. But how you're mixing and matching it for the individual patient therapy actually is uniquely personalized, but off the shelf now. This concept of antigen warehousing, I think, is one of the most profound and potentially impactful utilizations of the mRNA technology. Because now you can make something that's there and very quickly can be administered to patients and yet be very personalized to their cancer.
Okay. So maybe we can go to the other side of the lipid nanoparticles, right? So like, of course, I'll ask you to explain it, but like I'll try to explain it myself. One of the innovative parts that you've done is the way to introduce this genetic code in the treatment. And by that, I mean, like you use this very specific types of particles, like lipid nanoparticles, that have this specific geometrical structure so that you have many advantages with it, right? Like I'll let you do the explaining of the advantages and like why all of these things, like, right? Why lipid nanoparticles and like why the specific geometrical, and you'll explain like the structure that it has.
Yeah. So as amazing as mRNA is, it has one major weakness, which has really limited its development for a long, long, long time. And mRNA is not very stable. It can degrade very, very, very quickly. So it's very hard to deliver something that degrades so fast unless you have a delivery vehicle. The delivery vehicle of the information that is used in the COVID-19 vaccines, that we use in our mRNA therapies, they're not the same lipid vehicles, but they tend to be these lipid nanoparticles, lipid particles. Mm-hmm. Why do we use lipid particles? We use lipid particles because they are biodegradable. They're just, I'm oversimplifying, but fat droplets. Fat droplets that can protect the information. And because they're biocompatible. It's kind
of like their shield?
Basically a shield. Protects the information, and they can, because they're biocompatible, interact with cells, and the information can be transmitted to cells and program the human body in the manner that's desired.
Mm-hmm. What do they need shielding from?
So you have inside your body, you have something called RNA, outside the body as well, but you have something called RNases, basically enzymes that degrade RNA. Mm. So if you try to deliver, and people tried to do this once upon a time, try to deliver the RNA by itself, it'll degrade before it even reaches its destination.
Like you're sending something unusable, right? Like
basically.
Something that dies as soon as you put it in there.
Pretty much.
Like right, okay.
So that's why you need a delivery vehicle.
Right.
But there are advantages to these delivery vehicles. You can now build them to deliver information to the cells you want to deliver information to. You can build them to package more than one piece of information. So, when I think of nanoparticles, I think of, imagine a room this size. And you can put anything you want in that delivery vehicle. Right. Not just spike mRNA. Imagine you can load RNAs to turn on the immune response, turn off things you want to turn off, and do it all in one payload. So if information is power, the more information you can package and deliver, the more ability you have to leverage the information highway of the human body, the human immune system included, to achieve the desired outcome.
Would you say that the bigger, the better, in a way?
Yes and no. And the reason, and so you're getting into some of the challenges in the field. Mm-hmm. The bigger the better, yes, in the sense that you can package more information. But now, if it's so big, it may not even get into a cell because it gets stuck maybe in the blood vessels, and because it's too big to travel to the body and get into the right cells. So you have to be.
There's a sweet spot.
There is a sweet spot. There is a sweet spot. But you're right in the sense that I do think bigger can be better to a point. But look, nanoparticles have all sorts of advantages that can be leveraged. So if RNA can be used to make any piece of information you want, nanoparticle can be designed to deliver that information anywhere that you want. And I mean, to me, that's just very, very exciting.
Is there specific things that you can do with the nanoparticle to tell it to go to specific place that you want
absolutely.
it to go?
I mean, absolutely.
How does that work?
I mean, you can build these things really any way you want. You could put on the surface of the particle basically a targeting entity that tells that particle to go specifically to that cell with that type of surface target. So you can literally engineer these for that. You can, inside of them, you can put tracers inside of them to say, where's my vaccine going? Is it going to the right cells? Can I monitor the vaccine outside? If you put iron in the particle, can you now monitor it with magnetic resonance, MR imaging? Can you track where it's going? Can you? So people. I mean, I can go on and on, but you can build these really in a manner that's desired. One of the biggest challenges in brain tumors is the blood-brain barrier. Nanoparticles can help you get across the blood-brain barrier and deliver therapy into tumor. But this is not just for brain tumors. This can be for neurodegenerative diseases, Parkinson's, Alzheimer's. You name it. Nanoparticles give you an opportunity to access the brain, which has a very tight blood-brain barrier, which is why drugs don't get in very well, in a manner that allows you now to deliver care for diseases that otherwise don't have a lot of care. A lot of this is still very preliminary, but that is the potential of nanoparticle approaches. What we designed for cancer recently was it's not just what you have on the surface and it's not just what you have inside. It's the shapes of these things actually impact on how much product you can deliver and how the immune response will respond to something that looks dangerous. So we designed a shape that we think so we take the mRNA of cancer and we put it in an onion-like shape. So an onion allows you to add more material to a payload, a particle, because you have, instead of RNA is negatively charged, so you can't just, negative charged mRNA will repel.
Okay.
So we layer it, so you have RNA on the surface of a lipid particle, it's covered by another particle, another layer of RNA, and so it clusters. And so you have this kind of strong gamish of RNA that's in this kind of layered or multi-layered aggregate that we inject into the bloodstream. And so this is a very dangerous shape. So it's not just the amount of payload you can package, it's a dangerous shape. And we found this can very rapidly wake up the immune response in cancer patients.
Right, and it's the idea like you break a layer, deliver the mRNA, then it travels, breaks another layer, so like you can spread it over many distances, something like that.
So the truth be told, we're still trying to understand the biology of how this works. It's very complex biology as we're trying to understand. What we know right now is this can reprogram the ecosystem of the cancer that I described initially. It can reprogram the immune system in the rest of the body that's been kind of sleepy and not doing its job. And it leads to a multi-pronged attack against the cancer. And what we have to figure out, to be honest, is, how do you dose this? How do you boost this? How do you combine it potentially with other therapies to get maximal effect? And really understanding how it works is going to be critical to doing those things.
Right. Maybe could you speak a little bit on how these things are made? Like, there's probably, honestly, I have no idea. Right? Like, you're talking about this onion-like particles where you introduce the genetic code into all of these layers? Like, how do you even build something like that?
So when you're making these RNA nanoparticle aggregates, you have to make the RNA and you have to make the nanoparticles. And that's generally the field. So our lab has to have as much investment on the RNA side as it does the delivery side. And so when you're making these for patients, you need something referred to as a cGMP manufacturing facility. That's a scientific acronym for being able to make a human product in a controlled enough way that it will be allowed to be injected into a human.
Okay. Yeah. Okay.
And the University of Florida is a very special place because it's one of the few places in the world, honestly, that can make human-grade products like a pharmaceutical company and give those to patients.
And it just happens to be Florida, like the research labs and companies that live here have congregated. Or like, what is Florida special, like weather or something like that?
It's special because you have infrastructure here that most places
right.
don't have.
Right. Okay.
So
okay.
to be able to make something that goes into a human being, you need something called an FDA, Food and Drug Administration, investigational new drug application that has to be seen, reviewed, and approved by them before it goes into a human being. Now, in order to submit an application like that, you need to do small animal testing, large animal testing. You need to be able to do this testing under very controlled, in very controlled environments, something we refer to as GLP tox studies. The University of Florida, and I can't think of very many places that has all of this on one campus, has the ability to do small animal, has the ability to do large animal. And we've done this in pets, pet dogs and cats with cancer. We're able to test whether these things work or can help or are safe and feasible. Has the ability to do the GLP studies that I mentioned, pharmacokinetics and toxicity and safety, and you have to be able to formulate and compound these in a way that's going to be very similar to what's going to happen in a human being, and then you have to be able to manufacture it in a way that's quality assured, released to patients. And we have all of that ability here on this campus, which is really one of the remarkable things about the University of Florida. And in many ways, it has been the history of this place. When you think of what UF has meant to communities, it's largely been a place that has been a light for people, a hope for people where there is no hope, And this has been in the context of orphan diseases. So one of the reasons, I came here, our group came here, is because UF had all of this infrastructure that was being leveraged for genetic deficits, orphan diseases like muscular dystrophy, the invention of adeno-associated viral gene delivery technologies was really invented, developed, and moved into patients here at the University of Florida. We've leveraged that infrastructure for the development of cancer immunotherapies, personalized and universal cancer immunotherapies. But it requires this type of environment, these types of very complex manufacturing facilities to make these things.
Okay. And I'll definitely ask you about the whole FDA process of when you have something that you think works or again, early stages of the development of it, that you want to start testing it with small animals and big animals. Like, how does that process work? Like, in the sense of, like, how do you determine, for example, what has been working and and that is one question that I want to ask you, right? Like, in all of this process, when you're doing all of this testing with small animals, testing with big animals, testing with actual humans, how do you know that you're doing the correct thing? How do you know you're helping? And also the second part of it is, how do you know when you're not, right? Like, what possible consequences could come from this, and how do you know about them? Something like that.
Yeah, so, I mean, these questions you're asking are part of my my nightmare of
right
where we are, and I think where people often are when you're trying to do something for the first time. In many ways, I don't know that we do know, and that is frightening. I mean, we don't know what the right dose is. We don't know if this is going to hurt somebody. We don't know if this is going to do any good at all. We don't even know that it's feasible until you do it, right, for the first time. that's a hard place to be, especially when you're an oncologist and you want to do things that benefit people in a way that prevents much of the horror that I've seen. I mean, even when we cure cancer with chemotherapy, I mean, you can have a lot of side effects. So, the last thing you want is to be giving patients side effects or testing something that's just hurting someone. But unfortunately, that is a starting point for a lot of this, is that you just don't know. Now, you do the best you can. The FDA has a lot of guidance on here. If you're starting in small animals, this is how you go to a large animal. This is how you go from a large animal to a human. And then you drop the dose because you don't want to start at something that's even comparable. You want to start really, really low and then slowly work up to prevent some of the things I'm talking about. But human biology is complex.
Right.
And just because you treated dogs and just because you treated mice does not mean, and just because you have an idea of where you should start and you drop well below that, doesn't mean that there aren't going to be side effects. And I think this is often true when you're talking about biologics. These aren't drugs as much as they are biologics. Drugs, when we talk about drugs, these are agents that have to have what we refer to as the therapeutic index. Meaning, if you give too much, you're outside the index for effect, and you're getting toxicity. If you give too little, you're outside the index for effect, and you're not going to see. So, what is that nice window?
Right.
That's different for biologics often, because biologics, you're trying to manipulate the human body into doing something on its own. And how that self-amplifies may be unique to an individual, again, because all of our immune systems are very different, entirely personalized, just like disease. So if you give a biologic to a patient like an mRNA therapy, and it self-amplifies in that individual beautifully, and you get the perfect response, does that mean it's going to be the same thing in the next patient? Maybe they have a suboptimal response at the same dose. Or maybe they have an overactive immune response. Mm-hmm. And again, we designed something that's pretty dangerous looking and can I do worry about, in the sense of, the immune system is fire. And if this is a match,
interesting. Okay.
This could also be harmful if it's not controlled. How do you control it? You do a lot of what I'm saying. You have to make sure you go through the rigor, reproducibility, the manufacturing, the IND process, start really low, be very thoughtful about, not testing this in 100 patients simultaneously, but very thoughtful about slowly learning and using that information to do the most good you can for each patient. But I'll be honest, every time we treat a patient with these things, I'm terrified.
Right.
And it's when a biologic like this goes in the human body and tries to create a natural response, my fear is, what if this turns into a fire that can't be controlled?
Okay. How do you know? So, again, how do you know when you're doing good? Like, what is, for example, I was reading, I think, one of your papers in as best as I could, and one of the positives that came out of it, I'm not remembering if it's from the canines or the humans, is that the time, I think the survival time was greater with the patients that you administered this vaccine compared to the people that were not administering this treatment. I guess that's one of the ways that you can know. What are other ways to know that you're improving, like you're making progress, right?
Yeah, I mean, I think in the humans, it really remains to be determined. In animals, we've seen some remarkable effects, and some of this is unpublished. I mean, we've treated cats with, tumors that you could see, and we've seen those tumors melt away. And that is very, when you see that, it is very, it's a very gratifying.
So like one metric, for example, could be like amount of mass of cancer or something like that.
When you see it, it's extraordinarily gratifying.
Right.
The problem is, and we saw this in the dogs with brain cancer, we have noticed in dogs and humans that an inflammatory response in the brain looks bigger, not smaller.
Okay.
When you're doing the imaging.
Okay.
So when you have a tumor on the tongue of a cat, you could see the tumor, you could see it shrink.
Oh, interesting. Okay.
You can't see the tumor with your eyes when it's in the brain.
Right.
And when you look with a scan, an MRI as an example, what we're seeing in dogs and humans is often it looks bigger.
And the cancer or like the
You
something
can't
that-
really tell on the
Right,
MRI.
right,
It
right,
looks
right.
bigger.
Right.
Now,
right.
is it bigger because you have an immune response in the tumor and it's
Right.
swollen?
Right. Right.
Or is it truly that the cancer is bigger? And it's very hard to discern that from just an MRI that looks cloudy. What I can say, though, is these dogs have all lived longer than expected.
Right.
And the few human patients we've treated, we think are having better outcomes. It's impossible to say without randomized clinical control trials.
And some other metrics or like pieces of information that you can grab, I guess, would be something like whenever like amount of cell, like immune cells or something like that, or like what are ways?
So
to, like,
we've done that where we've looked at the blood of patients and we see that they're developing some nice immune responses and can
Okay,
respond against
right.
their tumor, but that's not a replacement, in my opinion, for outcome. Okay, are
Right,
they
and-
truly living longer?
Right. And
Are they-
outcome would be the living longer is the goal. Like, is that like the main driver? Like, you're trying for something that would most likely be deadly to some human or dog for that thing to live longer.
Well, the goal is cure. I mean, the goal, of course, is cure.
Okay, and like completely, yeah,
It-
exactly.
is
Like-
to eradicate
Right,
the
right.
disease. I think it's one of the things I've learned throughout this process is, maybe I'm just getting old, is it's a long arc.
Mm-hmm.
Meaning I can't just do everything I want to do in the beginning,
Mm-hmm.
because that could lead to a horrible reaction if you don't understand what you're doing.
Mm-hmm.
So you're starting at really low doses. You're giving few doses, and you're just trying to understand this so you can increase the dose, you can boost it, you can combine it with other agents, other immunotherapies that give you those curative effects. Along the way, I really want to see that, okay, this is helping people. They are living, even if it's not curing them, they are living longer. They don't have major side effects. These are things you really want to see as you're doing this, but it's slow. It's a long arc. But it is my hope that we will see this and we will start seeing these benefits in patients. And as we treat patients with tumors outside the brain, I'm hoping we'll see what we're seeing in animals, these things melting away. And that would be extraordinarily gratifying. But it is such a long arc to get to this elusive cure. And I'll be honest, I don't think this is, for some of these cancers are so aggressive and so evolved, it really is going to require a multimodal immunotherapy approach. Mm-hmm. So maybe not necessarily surgery, chemotherapy, radiation, or maybe with that, I don't know. But how do you combine immune agents? How do you combine T cells with mRNA therapies, with those drugs I mentioned, with other immunotherapies that are being developed and discovered? How can we use the immune system to light a match and cause a fire that melts that tumor away and allows the immune system to remember for life, protecting that patient for life, and yet not burn down the body in the process? And we're still trying to figure that out, to be honest, as a community. My hope is what we're doing will be used to help the larger community, will be used to help patients, and we'll get to that elusive cure. But I will say this, I do think it is likely personalized, meaning one patient may require T cells, vaccine, plus, minus, X, Y, or Z, and another patient may require a different combination. How do we predict that up front and rationally combine these things in a way that isn't us just doing stuff and risking a horrible reaction? How can we do something like that rigorously in a controlled setting that creates the impact we desire? And I think these are challenges that we have to figure out over the course of the next 15 to 20 years if we really want to make a dent in cancer outcomes.
What are the things that give you hope about the next 15, 20 years?
I have a lot of hope. I mean, I think as horrible as cancer has been, and I know a lot of patients feel like we've been doing this for decades and outcomes really haven't changed. there have been some incredible examples of response in certain cancers and certain disease. I mean, maybe it's not as widespread as people would like, but honestly, the way I see it is, these are drops in the water, and these ripples of success that we're seeing, I do think are going to lead to a tidal wave. I really do. I believe in it. I think it's immunotherapy. And that gives me hope that over the subsequent decades, that tidal wave is going to come for all cancer in some way, shape, or form. And while it may feel fleeting that, these small successes here and there, how is it really transforming things when you look at the investment that's been made in cancer research? I can understand why people, especially people who are personally affected by this horrible disease, I can understand why they may feel hopeless or feel like it's a lost cause. But I could say just in my time, I started in the immunotherapy research field when people said, people have been doing this for 150 years, nearly no success. Why would you spend your time doing this? I literally was there. And I don't think I'm that old. And in my time, in my 15 years of doing this, the field has completely transformed. If this is what can happen in immunotherapy over 15 years, what can happen over the next 25, 30, 40? So I'm full of hope.
Awesome. Maybe, how does all of this affect you personally? Because, for example, one of the reasons that I've seen other people stay away from medicine is just that it affects you personally. For example, not only like the work hours that you put into all of this and the stress which is there, there's a lot of work for sure, but like, for example, you dealing and talking and comforting like patients that are dying from all of this stuff. How do you see that? Like, what do you tell them? Like, how does that affect you?
I mean, that's a big question. It's not easy. I mean, this is being an oncologist has driven my entire professional career. I went back for my PhD. I went back into research because of my experiences as an oncologist. Seeing children die, seeing children suffer. It's hard. When you see a disease, I mean, to families, it's either 100% cure rate or zero. There is no 90, there is no 95. It's either you cure it or you don't. And when you have patients like I have who had a 90, 95% cure rate and you can't save them, it's an awful feeling. When you have patients who you cured and you could see the side effects of your therapy, that's hard. That's really hard when you see what it's done to them physically.
Right.
When you take a patient with a brain tumor and they can't walk, talk, hear after surgery, chemotherapy, radiation, it's hard. It's hard, and now they're living their life, and they can't drive, and they can't have kids, and they're cognitively impaired because you radiated a 4-year-old's brain with this very, very difficult radiation. So, I mean, that's really driven me into trying to find, new pathways, new avenues. And that's why I feel like I can't stop. But at the same time, I am scared. I mean, have I just shied away from seeing patients to pursue this research vision and replacing chemotherapy with something even more horrible? And the immune system has its risks, as we've talked about. And so those are some of the more hopeless moments because it's hard. There's a long arc to this journey. But I will say this, patients have inspired me and given me so much in my life. I mean, everything that we have achieved to date, whether it helps the patients we're directly treating or future patients, or hopefully both, that's been driven by patients. I mean, they have given me so much, and I just, in my heart, want to feel like I've given back something. So it's tremendously inspirational. It's hard at the same time when you see that level of human suffering. And how do you balance it? I don't know that I'm a great example of how do you balance it, because it's hard not to hug them and hold their hand and cry with them and not let that affect you personally.
Absolutely.
It's hard to go to funerals and see the horror that this disease has wrought upon families, and still maintain that objectivity I know we have to maintain. The way I've balanced it is through sublimation. I try to sublimate these feelings in our research, and hope that matters in some way more than the standard of cares we're providing in the clinic. But it's not easy, and I wouldn't say that I've done a great job or have found the right balance here.
What are, final, to wrap things up, what are you grateful for?
I mean, I'm grateful, honestly, for, most importantly, the patients, the families, for the privilege of being a part of their stories, to whatever extent that I have been, for their inspiration. I'm grateful for the beauty. There's so much beauty in oncology, and I know it sounds weird when you see horror, but I can talk about the beauty of patients who have brought divorced parents back together, patients who could inspire generations of medical students in the classroom, patients how much they did for me personally. I've had patients trying to comfort me when they knew that I wasn't able to help them anymore. Their strength is incredible. I'm so grateful to them. I've learned so much from them and their experience. I'm also grateful to the team of professionals. I'm one person, and I'm part of an incredible team of people. And I've said for a long time, we need to reproduce and research what we do in the clinic. I can't do anything for a patient in the clinic without nurses, social workers, child life, students, residents, fellows, specialists, consultants, surgeons, radiation oncologists. The team of people working together to try to save lives it's truly humbling. We need that same humility in research, and we need that same team model. And I've said this throughout almost my entire professional life. If we can work as hard as these kids and these families are working, I know we'll achieve great things.
Thank you, Elias.
Thank you. Cancer is not normal. And the immune response absolutely should have the versatility, the biology, to kill every cancer. And I would argue that it probably does, and we're just not aware of it.
Okay.
We're aware of its failures. But the idea that it can be resurrected in the fight against cancer is true.